this is undoubtedly the most valuable model, but every model has its limitations. Can you name a few challenges?
Yeah, sure. Like I said before, the primary use of the NKG mice is for its ability to engraft human cells. And the most common one is the human immune cell. So there are two major types. One is the primary PBMC, and the other one is the HSC, hematopoietic stem cell. So for PBMC, the use of NKG or similar is that because you are essentially putting in a mature T cell repertoire into the mouse, and then the T cells will recognize the mouse as foreign.
So the GvHD.
So the GvHD, graft versus host disease. The mature T cell is going to start attack the mouse, and then the mouse can become very sick.
So the shortened kind of window for experiment.
Right. So everything has to be done within 3 or 4 weeks. Otherwise, the mice will all die.
Yeah.
So it has a very short therapeutic window. And with the CD34 humanization, in theory, it should repopulate the entire immune cell repertoire because of the ability of HSC to differentiate into different cell types. But that's not the case inside of the mouse. And one of the reasons is that the mouse cytokine cannot support the fully development of human immune cell.
I see.
So that's two major drawbacks with the PBMC and the CD4 humanization.
Okay, so you can reconstitute the human T cells, B cells, NK cells, but not some of the other cells, especially the myeloid lineage cells.
Right. So myeloid is almost completely absent, and for B cells, you can see B cell markers, CD19, CD20, but they do not make functional antibody. They do not have the class switch IgG being produced.
Okay, I see. So in order to make improvement upon those limitations, people also, working on the so-called next generation NXG mice.
Yeah. So to circumvent the problems that I mentioned for PBMC, people trying to knock out mouse class 1 and class 2 because the GvHD is primarily acted upon the T cells, recognizing mouse MHC as foreign.
Of course.
So when you knock out the mouse MHC, then the mice survival is prolonged. And for the HSC humanization, because the mouse cannot crosstalk, the mouse cytokine cannot crosstalk with human immune cell. One way people to compensate for that is to make a transgenic mouse that express the human cytokines.
I see.
Like the example you mentioned, the MISTRG is an acronym for 7 different genetic manipulation, which includes 5 of the humanized cytokines being introduced into the mouse. But the problem is that you have to make the cross of 5 different lines to make the mice. And also, when you have so many different human cytokines, the mice can become very, very sick.
Yes. So do we have a solution?
Yeah, at Cyagen, we sort of take a different approach or path. Beyond, we are also making the next generation through the genetic approach. For example, the knockout of class 1 and class 2 for PBMC humanization, and also making, say, IL-15 NKG and the SGM3 NKG. But we also, in addition to that, we're also making a different route. Instead of using the genetic or transgenic approach, we use a AAV delivery system.
AAV delivery system.
Right. So what it essentially does is the human cytokines are packed into the AAV, and when the mice are still young, we use AAV to deliver the cytokines needed. So in this way, we don't have to maintain that much of a breeding colony. And it's a standard on the NKG, and also we can fine-tune the amount of the cytokines we give to the mice. And also because AAV is have a long-term expression, which we already validate in the serum from the mouse.
Okay, so you can reconstitute the immune system, in a much improved manner.
Right, and one thing stood out to me is to us is that we can actually get the functional dendritic cells being repopulated.
The DC cells.
The DC cells, yeah.
Okay, so the the antigen presenting cells.
Yeah, that was completely missing in the previous version. But we found the dendritic cells, the antigen presenting cells, being present in the what we call Pro-F.
Okay.
So the F stands for the full immune repertoire, and what we found that we have the increased dendritic cell by proportion, but also we can test its function in terms of cancer vaccine.
Yes, the cancer vaccine. Yeah, this is getting popular these days.
Yep.